Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning. Today we will examine the chemical reactions that underpin the photosynthesis process. We will follow the flow of energy from light capture through the light-dependent reactions to carbon fixation in the Calvin cycle. First, I will outline the organization of the chloroplast relevant to the chemistry. Light is harvested by chlorophyll in the thylakoid membranes, which are organized in stacks called grana. The thylakoid membrane houses photosystem II, an electron transport chain, and photosystem I. The compartment inside the thylakoid is the lumen, and the surrounding soluble phase is the stroma. The light reactions occur in the thylakoid membrane. Photons excite electrons in chlorophyll at photosystem II. Water is split at the oxygen, evolving complex producing molecular oxygen and protons. Electrons travel down the transport chain, producing a proton gradient that drives photophosphorylation. The light reactions therefore produce ATP and NADPH, which are chemical energy carriers. I want to emphasize the coupling between the light reactions and carbon chemistry. ATP and NADPH, generated at the membrane, are consumed in the Calvin cycle located in the stroma. In the Calvin cycle, CO2 is fixed into organic molecules. The key enzyme that catalyzes the first major step of carbon fixation is ribulose bisphosphate carboxylase oxygenase, commonly known as Rubisco. Rubisco catalyzes the reaction between CO2 and ribulose 1,5-bisphosphate to form two molecules of 3-phosphoglycerate. These three carbon intermediates are then reduced using NADPH and phosphorylated using ATP to produce glyceraldehyde 3-phosphate, which can be used to form sugars. An important chemical limitation is that Rubisco is relatively slow and it can react with O2, producing a costly process called photorespiration. Photorespiration returns some carbon back to CO2 and consumes ATP without producing sugars. When I mention that Rubisco is slow and prone to oxygenation, I want you to understand why plants evolved alternative carbon concentrating mechanisms. For example, C4 plants minimize photorespiration by spatially separating initial CO2 capture from the Calvin cycle. Mesophyll cells fix CO2 into 4 carbon acids, which are transported to bundle sheath cells, where CO2 is released at high concentration for Rubisco. CAM plants separate these steps temporarily to conserve water in arid environments. To make these ideas concrete, I will describe a classical experiment. Researchers used 14C, labeled carbon dioxide, and exposed green algae to light for milliseconds. By rapidly halting the reactions and analyzing intermediates, they identified 3-phosphoglycerate as the first stable product of CO2 fixation, revealing the sequence of reactions in the Calvin cycle. We can connect this chemistry to applications. In agriculture, understanding the biochemistry of photosynthesis guides breeding and engineering. For instance, transferring C4-like traits, or optimizing Rubisco specificity, is a strategy to increase crop yield in warm climates. In renewable energy, researchers attempt to mimic the light-driven electron transfer and carbon fixation steps to build artificial photosynthetic systems. Finally, consider environmental controls: light intensity, wavelength, CO2 concentration, and temperature affect reaction rates. High light increases electron flow, but if CO2 is limiting, then excess energy can cause photo damage. Stomato closure under drought reduces CO2 and increases photorespiration. These are chemical and physiological links that regulate overall photosynthetic efficiency. To summarize the core chemical sequence: light energy excites electrons in photosystems. Water is split producing oxygen. The electron transport chain generates ATP and reduces NADP+ to NADPH. In the Calvin cycle, CO2 is fixed by Rubisco and reduced to triose phosphates using ATP and NADPH. These reactions together define the photosynthesis process at the chemical level.
Listen again to part of the lecture, then answer the question.
When I mentioned that Rubisco is slow and prone to oxygenation, I want you to understand why plants evolved alternative carbon concentrating mechanisms.
This concludes the listening section. For more TOEFL practice tests and study materials, visit Spinta.org.